Hello everyone, welcome back to Botany class. In last video, we started the new chapter. So biotechnology, processes and principles, right? So where we discussed about the introduction part and what are the major steps involved in recombinant DNA technology, right? And in this video, we are going to see what are the tools we are going to use to manipulate the genome of an organism, right? The tools of recombinant DNA technology, right? And what are the tools we are having? Because here we are going to manipulate the genetic material of organism, because genetic materials are found inside the cells only, inside the cell within nucleus. So we have to, because they are molecules, they are microscopic one. If it is macroscopic, easily can manipulate it. But these molecules are the DNA, genes are, it's a microscopic one. So that to do manipulation, we need some tools. And what are the tools we are having? There are three important tools are available to manipulate the genome of organism. The first one, you can call it as restriction enzyme. So restriction enzyme also called as, it's a type of nuclease which cut the DNA molecule at specific site. The restriction enzyme also called as restriction endonuclease. And generally you can call it as molecular scissor or molecular knives also, right? Because we are using these enzyme to cut the DNA and to isolate the gene from the particular DNA, we are going to use this enzyme, right? That's why we are saying that molecular scissor. And next one we need for doing manipulation, because we want to isolate gene from one organism and we have to transfer the gene to the host, definite host. For that we need one carrier DNA. So carrier DNA molecule. The carrier DNA, you can call it as cloning vectors. So we need cloning vectors, and then the competent host. Host nothing but where we are going to transfer our gene of interest. That organism, you can say it host. Suppose I isolated the human protein encoding gene. So I want to clone this gene. I want to transfer this gene to the bacteria. There I want to produce human protein. So in these cases, where we are going to transfer now in bacteria, the bacteria act as a host. The host either may be prokaryotic organism like bacteria, or maybe eukaryote, either algae, fungi, or plants, or protozoan species, or even animals also. Okay. So where we are going to transfer that organism, we can call it as competent host. Okay. These are the three important tools. With the help of these tools, we are going to create genetically modified organism, nothing but recombinant organism, right? So these tools are available, right? We'll see one by one. First one, restriction enzymes, also called as restriction endonuclease. Under these restriction endonuclease are molecular scissor. So these are enzymes which cut DNA at specific region and make one linear molecule into fragments, DNA fragments. But these enzymes specifically may cut only double-stranded DNA, not single-stranded DNA mostly, right? So if lengthy DNA is there, the lengthy DNA, if you want to cut it into fragments, then with the help of this enzyme, you can cut and you make into fragments. And these enzymes are belongs to class nucleases. So nuclease is an enzyme which degrade nucleic acid molecule like DNA and RNA. So the restriction enzyme also comes under category nuclease. It's a type of nucleases which may cut the DNA at specific region or specific sites. Okay, see here, these are enzymes which cut DNA at specific site into fragments. Okay, see here one example they are given here. So there is a one enzyme EcoR1. It's a restriction enzyme. You see, these are DNA, double-stranded DNA molecule. It's making cut here and leaving two fragments like this, right? So this enzyme we are calling as restriction enzyme or restriction endonuclease. They comes under nucleases. And you see the type of restriction enzymes. Nucleases, there are two types of nucleases are there. Generally nucleases, so the enzyme which degrade nucleic acid molecule. So in cell there are two types of nucleic acids are there, DNA and RNA. DNA degrading nuclease, you can call it as deoxyribonuclease, DNase. RNA degrading enzyme, you can call it as ribonucleases. But here there are two types of nucleases are there, exonucleases and endonucleases. So what is the difference between exo and endo? Meaning is exonuclease and they remove nucleotide from end of the DNA, not at middle part. So not at any part. So only it may cut only at end. And also very important is, so this particular enzyme when they cut only one base pair at a time and mostly from end of the DNA. That's why we are saying that exo. Exona away or outside we will say. From both end it will make cut, but at the time it cut and relieve only one single base pair alone. That type of enzyme we are saying that exonuclease or endonuclease. So these enzymes in the cut DNA molecule at any part other than end, at any part other than end. But where it will cut, not specific position, at specific part or specific region, it will make cut within the DNA molecule. That enzyme we are calling as endonuclease. Okay, see, exonuclease cutting DNA only at end, but endonuclease cutting DNA at middle within DNA, not at the end. So that's why, but where it will make cut na, so they are having some at specific places it may cut. So you see, these enzymes that bind to specific regonation sequences of the DNA and cut the two strands at specific point and leave DNA fragments. Okay, see here, the image they are given. The enzyme cut both DNA strand at same site. Some enzyme they will cut like this. So here and ECOR1 cut on the we are having the DNA, foreign DNA and two different DNA. So we are making cut using ECOR1. So ECOR1 what will cut and remove DNA fragments. Sorry, generate DNA fragments, right? And we'll come just to give, we'll see the historical event. In 1963, two enzymes responsible for restricting growth of bacteriophage in E. coli. The bacteriophage, we know that it's a virus, it's a DNA virus that specifically infect bacteria. So that's why this virus we are calling as bacteriophage. And the scientists, they isolated two enzymes responsible for restricting growth of bacteriophage in E. coli. So in E. coli, these enzymes are isolated from E. coli bacteria. These enzymes specifically restrict the growth of bacteriophage. When the virus infect entering into E. coli, that time the growth of bacteriophage may be restricted by or prevented by so two enzymes first they isolated. So how these enzymes they are restricting the growth of bacteriophage now. One enzyme add methyl group to DNA. So at the end part. So what will happen is if methyl group is added to DNA and then automatically which prevented from the action of nuclease. And other enzyme what will make na, it may cut the DNA of bacteriophage. So you see, so one enzyme adding DNA, actually you see here when cell is entered, okay, see for example you take here bacteria, this is a bacteria, so inside bacterial DNA is there, right? And here this bacteriophage also inserting its DNA, right? So now you can see here. So just I'm taking one bacteria, right? This is a bacteria. The bacteria is a bacterial DNA. So this bacteriophage after infection, So this bacteriophage also, it will inject DNA into the bacterial cells. Okay, this is a viral DNA, this is a bacterial DNA. So these cells, okay, these cells alone contain two enzymes. So one enzyme what will do now, they will add methyl group here. They add methyl group. Here they will add methyl group, right? Methyl group they will add to the bacterial DNA. And another enzyme what they will know, they cut and remove this viral bacteriophage DNA from these cells. So these enzymes are, one is a methylase, another enzyme name is called, the enzyme which make cut name is called restriction endonuclease. So these two enzymes only they first discovered from the E. coli bacteria, right? Because one is they want to cut and degrade the viral DNA, and at the same time they want to prevent so that own DNA from the action of restriction endonuclease. Okay, so one is methylase, another one is restriction endonuclease. And up to date, so more than 900 restriction endonuclease. So more than 900 different type of restriction enzymes were isolated over 230 strains of bacteria. And also the restriction enzymes are found only in prokaryote, not in eukaryote. Prokaryote like bacteria, not in the eukaryotic organism. So up to date, the scientists isolated 900 different type of restriction enzymes from 230 different strains of bacteria they isolated, because these enzymes found only in bacteria, not in eukaryotic organism, right? Actually, how we are going to assign name to restriction endonuclease or restriction enzyme, the nomenclature of restriction enzyme. For this example, here I have taken EcoR1, very commonly used enzymes in a gene cloning experiments or genetic engineering experiment, EcoR1. So EcoR, when we are writing, they'll start with caps letter and then small letter and then abbreviated form, some extension will be there given, right? So Eco, the first letter indicate genus. So indicate the genus, E. coli, Escherichia coli, we are saying that. So first letter will indicate genus name. Second letter on the second two letter on the indicate the second two letter with the species name. So there is a prokaryotic cell from which they are isolated. And R is the strain. So in E. coli there are different strains are there. Strains nothing but, so all are belongs to same species but genetically somewhat different organism. And I is, it's a Roman letter, the order in which the enzymes were isolated from the same strain. So the same bacterial strains, maybe the scientists they isolated more number of restriction endonuclease. In that, the very first enzyme is EcoR1. That's why they are given Roman letter 1, right? So EcoR1 comes from E. coli RY. The strain name is RY. That's why they are given RY13. From that organism, they isolated. So that's why they are given name as a EcoR1. Like similarly, Hind3, BOMH1, like there are many enzymes that we are going to see that examples. So like this, the first letter on the which denotes, which indicate the genus, second two letter on the species, third letter on the which strain they are isolated, and further on the Roman letter they will use the serial number of the enzyme. From same strain they may isolated more number of enzyme. Among that the serial number they are given as a Roman letter. So EcoR1 is restriction enzyme isolated from E. coli strain RY13. From that the first enzyme is EcoR1, right? Next we can see the first restriction endonuclease discovered is. So which enzyme the first discovered? The very first discovered restriction endonuclease is Hind2. Keep it in mind. The very first discovered enzyme is Hind2. But this enzyme cut DNA at specific region or specific sequence. The specific sequence you can call it as regonation sequence. Mostly the sequence are they are 6 base pair long. The regonation sequence for restriction endonuclease are they are 6 base pair long. So that particular sequence name is called, you can call it as a regonation sequence for restriction endonuclease. Here they are given the regonation sequence for Hind2 because enzyme all restriction endonuclease, they are having specific regulation sequence. The regulation sequence may be vary from enzyme to enzyme, right? Enzyme to enzyme. Here they are given, you can see Hind3, Hind2 regulation sequence. Okay, GT, pyrimidine base, purine, adenine, and guanine. And CA, purine, pyrimidine, and adenine, and guanine. Actually, the restriction sites, the base pair, how much base pair long? Na? Minimum, they are 4 base pair long, and the maximum it goes up to 6 base pair long. 6 base pair long. Here they are given the regulation site for various type of restriction endonuclease, like ALU1. For this restriction enzyme AGCT, the base pair, and HAE3, like you see GCCC, and BOM H1. So these are all actually HAE in a hemophilus, hemophilus amylosensine. Like BOM H1 na Bacillus amyloliquefans. Like and Hind3 na Haemophilus influenzae. So for this and all, only these particular enzyme they can make cut if the DNA contain the specific sites for that particular enzyme. Okay, you see BOM H1 when it will cut na, the DNA contain these sequence only. These are specific sequence for only for this particular enzyme. Like Hind3 also, like AA, GC, TT. So we will make cut only the particular sequence. Like ECOR1 you can see GAA, TTC. Okay, so when we are cutting this DNA, it produces DNA fragment with 2 types of end. One is blended, also called as blended, or you can say flush end also, or plain end. Another one produces sticky ends. Sticky ends also called as coccasive or coccasiones also, with single strand overhangs. Okay, at both end contain the stretch of single strand will be there. That's why we are saying that sticky ends. So next one is restriction. The types we will see, we are discussing here. And I told restriction enzyme, they can recognize and cut the DNA only at specific sequence. The specific sequence we called as a recognition sequence. But all recognition sequence of restriction endonuclease are, they are palindromic one. They are palindromic nucleotide. Palindromic meaning is the sequence of base pair that read same on 2 strands. Like when we are reading one strand 5' to 3', and another strand 3' to 5', it's a read in same. For example, you can take Malayalam. Malayalam, if you read it from M-A-L-A-Y-A-M. In reverse also M-A-L-A, and Madam, M-A-D-A-M. Like reverse also you can read. That sequence you can call it as palindromic one. So the recognition sequence for restriction endonuclease, all are, it's a palindromic one. Okay, see here example. So 5' to 3', G-A-A-T-T-C. When you are reading this direction in this stand, G-A-A-T-T-C. Similarly for all sequence. For example, ECOR1 you can see this is an ECOR1. The Hind3 you can see A-A-G-C-T-T. If you reverse like this, A-A-G-C-T-T. Because this sequence you can call it as palindromic sequence, right? Under these restriction enzymes, they cut DNA molecule, DNA strand little away from the center of palindromic sites. So little away from the center of palindromic sites. But in between the same and 2 bases on the opposite strand. And when we are cutting little away from center of palindromic, which leaves single strand overhang stretches at the end. So these ends are, you can call it as sticky ends, right? You see here, it's a making cut here. GAATTC is there. Here the enzyme make cut here and here also. When generating DNA fragment, which generate with, so which leave single strand overhang. So at the end. So this end you can see here up to GC here is there. But here only bases are there, but there is no base pair. it's cutting little away from the center. Okay, you see the ECOR1 restriction sites are GAA, TTC. But cutting middle part is only these part is considered as a middle. But cutting little away from the center, from it making cut on this stand here, and here in on this stand making cut here, so that leaving DNA fragment with a single strand overhang. So this end you can call it as sticky ends, right? So on the sticky ends, why? What is the advantage of the DNA fragment sticky ends? So when we are inserting this gene into the vector, that time the sticky end form hydrogen bond with the complementary cut counterpart. So the stickiness facilitate action of DNA ligase. So when cut by the same restriction enzyme, the DNA, the fragments have same kind of sticky ends, and it's easy to insert and ligate and join using DNA ligase. Okay, see here vector DNA they are given. Here same ECOR1 sites are there. Here also same. When we are making cut with this both DNA using ECOR1, it generating these fragments generated like sticky ends, and this one generating sticky end. Here you can see this end contain TTAA, but here AATT. When you put together easily, it's a complementary basis. Easily they can form hydrogen bond, so that these fragments are joined with the help of DNA ligase enzyme. So for that, so most of the enzyme they produces sticky ends. Only few restriction enzymes they produces blunt end, right? And so you see another example they are given. The foreign DNA here we are taken vector DNA, and we are cutting this DNA at specific point using restriction endonuclease. The vector also same enzyme we can use to cut the vector DNA also. Now the piece of foreign DNA is here inserted and ligated using DNA ligase. Now this molecule, because this is a vector, this is a foreign gene, both the foreign gene is inserted into this DNA. Now this DNA can call it as recombinant DNA molecule, and this recombinant DNA transformed into the host cells, and then host cells allowed to multiply. So that the host cell will transfer it the DNA to the progeny organism, right? So far we discussed about restriction endonuclease enzymes, right? And the next tools are cloning vectors. Cloning vector. What is that cloning vector? It's a DNA molecule that can carry foreign DNA segment or replicate inside the host cells. So the cloning vectors are nothing but they are DNA molecules only. Definitely they are double stranded DNA, which carry piece of foreign DNA from different source, alien piece of DNA, and they carry and transfer this into the host, and also they can replicate inside the host cells. The most commonly used vector, which vector we will use now? Plasmid. Already we discussed. So the plasmid DNA and DNA of bacteriophages, because all the bacteriophages, the genome is double stranded DNA. So that plasmids and bacteriophage DNA we are going to use as a cloning vector. So the plasmids, we know that plasmids are autonomously self-replicating extra chromosomal DNA found in bacteria. Okay, see here bacterial cells, this is a chromosomal DNA responsible for heredity of this organism. But the same cells contain some extra piece of the DNA. In addition to chromosomal contain extra DNA molecule. So these are circular, double-stranded, autonomously replicating, self-replicating molecule. This molecule we are calling as plasmids. But as bacterial cell, how many plasmids are there? Some plasmid they have 1 to 2 copies per cell. But some organism they may have 15 to 100 copies per cells also. 15 to 100 copy per cells also. And another one, bacteriophage. The high number per cells have very high copy number of their genome within the bacterial cells, because one virus, if infecting, it inject DNA, the DNA, bacterial DNA multiplies so faster. So the more amount will multiply. So that we are going to use bacteriophage DNA also as a vector for cloning in prokaryote. The plasmid and bacteriophage are suitable. It's a DNA, both DNAs are suitable vector for cloning in prokaryote, right? And next you see, when the cloning vector are multiplied in the host, the linked piece of DNA also multiplied to the number equal to. Okay, see, I transfer one the rDNA molecule here. When this molecule, when multiply within these cells, 1 become 2, 2 become 4, then the whatever the gene we inserted in within this DNA also multiply. The numbers also they will multiply, right? And next we will see the vector for cloning genes in plants and animal. So plasmid bacteriophages are suitable for cloning in prokaryotes. And what about the plants and animals? And the plants, we are having many number of vectors. The very important vector is we are going to use some pathogenic DNA as a vector for delivering gene into the plants and animals. For example, for plant, the Agrobacterium tumefaciens are there. Agrobacterium is it's a gram-negative rod-shaped bacteria which specifically infect the dicot plant and which cause ground gall disease, right? The tumor-inducing, they're having tumor-inducing plasmid. We are going to use this plasmid, the plasmid DNA from Agrobacterium tumefaciens as a vector to transfer gene in plants, right? Agrobacterium tumefaciens. And also for animal, most of the animals are affected by retrovirus. So retrovirus, they are having single-stranded RNA as a genome, so that they specifically infect animals, mammals. So that for cloning in animals, we are going to use the retroviral RNA as a vector. From that, you can prepare cDNA, and you can use that cDNA for delivering gene into the particular organism, right? I hope that you understood. So up to this, we discussed about the restriction endonuclease enzyme and what are the different type of cloning vectors we are having, okay? So next class, we'll continue what are the feature required for cloning into vector. Suppose one DNA molecule, I listed the different DNA molecule act as a vector. If one DNA, if we want to use the DNA as a vector, and then what are the features are required? Just you see here, origin of replication, selectable marker, cloning site, and vector for cloning genes in plants and animals, right? So next video, we'll continue the features of the cloning vectors, right? And thank you for listening. We'll see next video.